We're now beginning. So before we get started, I'm doing it, I'm saving my voice. So I've done two relatively brief recordings, I think maybe 24 minutes total. Just did one this morning, I did one late last night after the game. And I converted them to YouTubes and sent them to you. Listen to those, okay? I'm sitting there looking at the test on my computer, talking. So whatever's on there, that's what's following. Again, I get tired of people asking me when the test was. October 1st. Okay, so yeah, you drive me sugar. That's crazy in Yiddish. Nevertheless, so and again, you have the advantage here. By the way, so I'll reinforce this point. You're the first group. You have a subtle advantage in the sense that I'm going to walk in here at 20 to 9. If you want to be here early, I know we have sometimes a late arriving crowd here. I like to call fashionably late, but that's okay. So you get a little extra time to start on that end, and as you filter out, the other class I'll tell them they can come in. So the hard time is toward the end, but we'll go from there. Nevertheless, that's the mechanics of the day so far. Listen to those, listen to the other recordings, look at the study guide. I know it's a novel idea to send me an email, ask me a question about some pertinent information, but you should try it sometimes. Or even ask a question in class. It would amuse me. But I digress. Very good. We're moving on. We did simply diffusing. Now we're going to be a little more complicated. We're going to be facilitating diffusion, and we're going to get on finally to active transport. So moving on after osmosis. So that is this particular PowerPoint here. So again, facilitated just means you have something that enables or permits. You have a protein channel that's there, a passageway, a doorway, any way you want to think of it. So, and by definition, these are molecules that are, glucose, hydrophobic. That's incorrect. These are, you should say hydrophilic, another wonderful PowerPoint error. Glucose amino acids, ions are transported passively. The idea of diffusion, again, is using the concentration gradient. Typically they go through some type of carrier, but if the carrier, the distinction between channel and carrier is channels are continuously open, and carriers typically have something that attaches to them, or they're enabled in some way, shape, or form to allow things to move. I'm not going to get into a big discussion about that as well. So integral proteins is what they are. They carry specific polar molecules, so now they got it right, that are too large for the typical membrane channels that are there. Typically, the channels are more or less designed for water, and as you will see, you're going to see, if we get a little farther on, start talking about resting membrane potential, you're going to see sodium and potassium charged particles that have channels of their own. You'll see the name in a second, called leakage channels. So when something binds to them or enables them, it allows it to change shape, to envelop it and bring it in. And the whole limiting factor is you only have X number. That's less of a problem for us than what we're going to be studying. This idea of saturated carriers, that's more classic anatomy and physiology. I don't get too excited about it. Neat pictures. Okay. This is the difference between channels are interesting, and it's sort of an overlap, because when you have a gated channel, effectively, that's a channel that's not always open. So that's why I don't like to go into carriers and channels. But the distinction you will see are these channels are going to be really important down the road. Okay, channels formed by these proteins. They transport the molecules, ions, water down the concentration gradient. It has a lot to do with the different factors, the size, the nature of the charge. When it's water, we use the term pores or aquapores. And this is the difference between leakage and gated channels. Leakage channels are always open. Gated channels, and they're controlled. As you will see when we talk about chemical gated channels, you'll see the term ligand, term that I put on the board before, ligand gated channels. Something opens that channel, it permits it. And the other you're going to hear about a lot, because it's how nerves and muscles work, voltage gated channels. You're going to see both of these involved in it when we get into the intricacies of muscles and nerves in the second half of the course. be happy when we're done with this. We're going to bones when we finally get through all this cell stuff. I'm ecstatic. Bones, you guys are ecstatic for bones. Is he going to have open lab? Is he going to have bones out this today? But I know, but is he going to have bones for you to review? Because I have a lab at 3 o'clock. So, I mean, I need the bones then because I'm doing bones also. You're welcome to sit in and listen. Got nothing to do at 3 o'clock, bring a snack. No food or drinking in the lab. Okay. Small lipid insoluble, there's channels. Osmosis, movement of water, always confusing to students. Shouldn't be. All about particles. So, I mean, think, imaginarily, you have two containers. They're equal in volume. You have particles, and I'll just do numbers, it's easier than doing pluses and minuses. 7, 8, 9 here, these are equal in volume, 1 unit of volume. Here you have 1, 2, 3, let's say. So here you have 9 particles in a container. Here you have 3 particles in a container. Except the container is filled, both of them are filled with good old H2O, water. So we're always talking about solutes to solvents, but they both are involved in concentration gradient. We're always thinking about sodium is moving this way to test. What about the water? You always have water channels open. All you have to do, if you want to visualize it, you flip the fraction. Just by looking at it, there's more water here than there is here, proportionally, when you look at it. So if you flip it, this becomes 1 unit of water over 9 particles, 1 over 3 particles. I don't know where you come from, 1/3 is bigger than 1/9. That means there's more water here. So if this magic doorway would open, that would allow, where do we go this way? You have to understand that a lot of times it depends on the opening. If it's big enough for everything to move, they'll balance each other out. But if it's not, water is the part that can always, with almost no exceptions, but with very, very, very limited exceptions, water can move easily in between cells or from cell to cell or from unit to unit. So movement of the solvent by definition is going to be water, selectively permeable. Even though water is polar, it is so small, a lot of times it can get by. Okay, or it has the aquaporins. And again, other than having those tight junctions permitting the flow in between the cells, that's different. Tight junctions are about where the cells are joined together, not this kind of thing. This way water is literally moving through. Okay, and so it's just like any other item. Whether there's more sodium on the outside than inside, sodium's going to flow in. Doesn't matter. Water plays the same game. And so they're just showing it to you, whether it's going through a channel. This is the fancy term you don't have to know. Okay, osmolarity. It has to do with part that's so important biologically, and what we do, we know roughly in our bodies. Just go with the number. You don't have to remember it now. It's called 300 milli. Osmoles. Okay, that's the relationship in our body of all the solutes. And again, the neat thing about that, it doesn't play identically. So even though salt is very tiny, sodium and chloride, and let's say sugar, glucose is quite a bit bigger. When we put glucose in water, it dissolves to have 1 unit. When we put sodium and chloride, it has 2. So size doesn't matter in this particular case. It dissolves into a sodium and a chloride or an iodide. That's why, and we add up all the particles, whether it ionizes 2, 3, or 4 particles, whether it's a protein, whether it's a sugar, whether it's a different sugar. We add up all the things that are dissolved in water. That's why I did put it. I just use numbers to represent that. That's why we look at osmolarity. It's all the stuff that's in there. And I'm going to show you in a second. So different osmolarities are separated by a membrane permeable to all molecules. Basically, everything moves to balance itself out, including the water, until it reaches what we call an equilibrium. That means if there's glucose over here, 2 over here, or 3 over here, One will move this way. Oh, I'll keep moving that way until roughly a balance is established. And you have a little one moving here just because molecules move. If they're not moving, guess what? You're dead. I say dead a lot. Forgive me. That whole pass thing, that's what you do with a test if you pass. I know people don't like to use the dead thing, you know. I hear you. It's okay. So I'm sorry to be politically incorrect. Same concentration of solutes and water molecules on both sides with equal volume. So here's this magic membrane. So it doesn't matter that there's more solutes here. They're doing it with sugars. If it's freely permeable, they'll just balance each other out. And equilibrium will have one moving this way and one moving that way, and the water moving this way and the water moving that way, because it's not static. But if we take the same illustration, permeable only to water, only osmosis, not diffusion occurs. So the water balances out. This happens all the time. So water will move to balance out the appropriate ratio of solute to solvent. And so effectively, here's this magic membrane. We open the door, and voila, the water has to lower here and raise here. Happens all the time, biologically. The whole idea, and what this dovetails into, and that's the most important part of this, is because of that 300 milliosmoles, we know that there are certain solutions we're infusing intravenously. So careful, because if you would start an IV with pure water, kill somebody. We never do. Typically, use 0.9% sodium chloride. We call that normal saline. Fancy name for these solutions is isotonic. The tonicity, that word tonic means the solute to solvent ratio. Iso means same. So the same tonicity or the same ratio or the same molarity, okay, of that solution makes it normal as to the plasma, as to the extracellular, as to the intracellular fluid. So if we take 0.9 grams, 100 milliliters of water, so each salt of water, not like seawater, okay, and certainly not like plain water, and that's what we sterilize and typically start an intravenous with. There's almost no fault, unless there's special conditions. Why we do that is it's neither going to move water in one direction or the other. If we have a cell, doesn't have, it's not like a plant. Plants have a cell wall. If you put extra water in them, what happens if your plant's a little droopy? You need water. Like water, I'm feeling good about it. It works up. I'm happy. Not us, you'll blow our cells up. If you put salt water into the plant, it's going to pull the water out. It's going to shrivel and die. Okay. I have a deck that's filled with plants. Okay. And also veg plants. Our peppers are still growing. Yes. Okay. I'm green with envy about the green. Just shut up. Nobody cares about your peppers. We don't grow it unless we can. That's the way we roll in our house. Okay? That's the whole idea. So, what are other solutions that mimic this? Years ago, I don't see it used as much. Again, 25 years since I ordered an IV. Okay, this is called D5W, dextrose. Again, the active aspect of glucose, 5%. Water, it's named D5W. What you do see used a lot, particularly in burn patients, which are trying to replenish salts, sugars, and a variety of chemicals, called lactated Ringer's solution. You'll hear that name if you're going into a clinical field. All of these are isotonic medium. You see this again and again. And undoubtedly they show you the same illustrations, and I've done these experiments. So what does this do? Okay, hydrostatic pressure is your blood pressure. Okay, it's not just pushing fluid that way. I did this before. It's not pushing it down a blood vessel, it's pushing it against the walls. When they measure your blood pressure, it's the force against the walls of the pressure. You squeeze that cuff, when you begin to release it, when those walls are being pushed back out. That's the start. We call that the systolic. Okay? So it's basically the cells are being pushed, all of these types of things that are there. Everything that's holding that fluid in is called osmotic pressure. The fancy name is colloid, a term we used before. The proteins principally are holding on to the water. It's a tug of war. Blood pressure pushing it out, osmotic pressure holding it in. And as long as it's kept in balance, there'll be a sufficient amount of pressure differential to push fluids out when it gets to the capillaries at the cellular level and bring fluids back in. And you'll see it when you get to blood vessels. This is an AP2 concept at the start of a capillary bed and at the end, because that's where the exchange is. You'll get to that. Okay. So the more solids in the cell, the bigger the pull on the water to enter. That's osmotic pressure. It's not just solutes per se, but that's a good. Mostly the proteins play a role in that. So water trying to get out equals water trying to get in. Plant cells, the strong cell wall, act to limit that. That's why they're very resistant. That's why bacteria, which for all intents and purposes, have a very strong cell wall, using a lot of water is not going to kill them. It's amazing. Most of them can't tolerate salt at all. We've been using salt forever to control bacterial growth, even in the Roman Empire. Did I tell you this one? Probably not. Okay. How did they pay the Roman soldiers? Salt. That's the derivation of the word salary. They used the salt to pack meats and pack vegetables. It would suck water out of it. Bacteria couldn't grow, so it was preserved. I mean, they didn't have a fridge back then. They'd have to go to the polar areas and not polar, but to the mountainous areas and get ice and things like that if they did need to have that. Okay? And that's where the expression came. He wasn't worth his salt. And then after that, if they didn't do a good job, they were going to you guessed it, salt mines. See, isn't it good? History's your friend for studying science. So too much water, it all bursts, shrink, cells volume can disrupt cell function. The cell is an environment that requires a very consistent relationship of solute to solvent. So again, these are the big terms to recall. And this is all about osmosis. Isotonic, the same osmolarity as inside as outside the cell. Hypertonic, higher. And the fancy term for this is called crenation. When it crenates, it shrinks, shrivels. And if it's hypotonic, pure water, deionized water, triple distilled water, okay, it pushes it in and the cell will lyse. Another term we often use for that, you'll see different terms, typically we call it plasmolysis, because it's lysis of the plasma or cell membrane. Lysis means split or burst. Let me show you the illustrations here. The sodium is 2, osmolarity, don't care. I used to do this in micro. Play with a microscope, and I did it in my physiology lab. I probably will do it this year when I do the advanced phys, because we usually get some blood, and you can use basically blood that they've rendered not harmful to you, so you can't get any blood-borne disorder. If you put red cells, a tiny amount of red cells with normal saline, they don't change at all. You're able to see the classic look microscopically. Okay, if you put it with a little bit of salt water, it won't. This is a micrograph, an electron microscope. You'll see points start to form. Basically, this will start to square off and form some type of a pointed geometric figure. Obviously, that's something that's not going to be nearly as functional. It bursts so fast. I am really fast at focusing a microscope. I got my first microscope how old? I was that kid. I think 6. See, my parents, after I got the chemistry set at 5 in the kitchen, they just said, we'll go with the microscope, it's safer. They had a little alcohol lamp instead of a Bunsen burner. I didn't know. So little damage at 15 Briar Hill Circle in Springfield, New Jersey occurred. I was that kid. Moving on. So the whole idea, if I try to focus it super fast, I can't find it. They burst that fast. If you had some way to look at it continuously in real time, maybe you would see them get to this. They're all gone by the time I focus it. Okay, save the animation if you want to look at it, voila. That, ladies and gentlemen, was, as we say, passive movement of membranes, and terribly important because of the osmosis as much as anything else. Now we get to something entirely different, active transport. Two types. And now you're going to see something about pumps. We have active transport and what's called vesicular transport. When we start talking about endocytosis and phago and pino and exocytosis, anytime you open and close membranes, even though they're inherently, there's a degree of stability, you require energy to do that. That's how we bring bulk things into the cell and push bulk things out. First, active transport, effectively moving molecules against the concentration gradient. Either it's too big, and that's when we use bulk transport. It doesn't get through the membrane, as most of these are, and it's not able to move down its concentration gradient. In order to somehow get that into the cell or expel it from the cell, we must impart energy, spend some ATP. You'll see typically when we spend ATP, as we create a push outflow or a push inflow, we can move things along with that current, so to speak, or even use that current in reverse to push things out. The fancy terms you'll learn about called co-transport and anti-transport. Not so much important for now. Down the road, depending on who's giving you AP2, that will give you, they may or may not cover that. That's there. Here they are. So some carriers transport, again, conserving the amount, maximizing the energy. So anti-porter, the opening that doorway, okay, we can move something in and use that energy and push something else out against this gradient. We see that all the time. That's there. The classic way sim porters, sim or co, similar transporters, are both moving stuff in. Typically when sodium comes in the kidney, it brings glucose along for the party. So you don't want to, you want to retain your glucose. Diabetics, when their glucose levels get too high in their bloodstream, you can't bring it all back in and conserve it. You're going to spill it out into the urine. That's really what the hallmark of diabetes has always been, spilling sugar into the urine. When people started to do that in the old days, we didn't have those neat little finger stick things. You basically dipstick the urine to see if there was sugar. That's how you basically adjusted their insulin level that were there. I mean, the finger sticks have only been around less than 50 years. Less than 50 years. They were somewhat in their infancy when I started to learn about this stuff. Okay, so those are the terms. I'm not going to make a big deal about those terms. I just want you to understand for the time being. If I need you to know them, you'll know about it. ATP. So primary active transport comes directly from the ATP. That's symporting and antiporting, that's indirectly from the ionic gradients that occur. So effectively, it's like somebody drafting in a pool or in a racetrack, in car racing, that type of thing, using that energy, which makes it easier for you to move things in or move things out. If that's an analogy that works for you, that's fine. Primary active transport is something you'll see very, very shortly. Secondary is something you begin to see when you look at the systems. So primary causes the change in shape of that protein. Okay, and the best example are these pumps. When we get to digestive, when you get to that in AP2, you'll learn about proton pumps. That's how the stomach manufactures acidity. There are pumps for calcium as well. The one you see nearly in every single cell is called the sodium-potassium pump. It's the one that is best studied by far and away. Fancy name for it is really an enzyme. Okay? More than anything else, it's called sodium-potassium ATPase. you're going to see in our cells, we're constantly trying to maintain something called the resting membrane potential, which I've touched on before. It pumps sodium's coming in, potassium is effectively what happens. Let me rephrase it. In our cells, there's always more sodium on the outside of the cell and more potassium on the inside of the cell. It's very, very critical along the cell membrane. You'll see the animation, which hopefully is coming up very shortly. All plasma membranes, but particularly nerve and muscle cells. A typical cell, one of those epithelial lining cells that you studied in lab, okay, whether it's simple squamous or not, they all have a resting membrane potential. They don't use it. It has to be a cell that has the ability in that membrane to basically, it's almost like waves going in a pool. If you throw a rock into a pond and you watch the wave, that's basically what's happening. Those cells have the remarkable ability to conduct an electrical impulse along the cell. That's when we get to the nerve and muscle. It's the basis of physiology. So let me do it this way, because there's a lot of stuff. And all that stuff is, I don't know about you, it's confusing. Too much. Remember I told you about good animations and bad animations? Now we get to good animations. Let's us see.
About 100 billion nerve cells, all neuron.
Go for it.
The human brain alone contains about 100 billion nerve cells, all neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
And you can view this as often as you wish on this. That's why I like to use this PowerPoint sequence. Effectively, all of our cells have that. Fortunately, most of them are relatively stable, and so we don't have to spend that energy by way of those sodium potassium pumps. Most people will tell you about half of your energy every day goes to those pumps in nerve and muscle cells. An immense amount of ATP is expended there. They've been studied for people when they talk about chronic fatigue syndrome and other things, whether it's not this particular pump But now you're getting your real first view into understanding really about physiology. We've had a lot of terminology, but understand every single cell we talk about has a polarity around the membrane. It's relatively neutral, the fluid on the outside. So if we take a generic membrane like that, you're always going to be more positive on the outside than on the inside. Understand that you have neutrality here and neutrality inside the cell. It's balanced out. And you're always going to have more sodiums on the outside and more potassiums on the inside. The reason why we see this consistently is because we have a greater number of these are leak channels. Pure and simple. You have a greater number of potassium channels leaking. So the key player in all of this is potassium. You've already heard me say it a couple of times. Doctors get very excited. Nurses get very excited. People in clinical fields, when people's potassium theories, charged particles, sodium, there's a great deal of abundance. So it's very rare that you see irregularities in sodium. That's a fairly wide range. Not so for potassium and calcium. There's a very, very close range about those. Looking at them all the time. Potassium is the big player because of all those leak channels. And if there's any degree of activity in the cell, even just basic mitotic activity, it's going to alter that relationship, and the sodium-potassium pumps are going to be busy doing that, and nerve and muscle cells all the time. That's why I get away with saying I'm so tired, because I'm thinking all the time. See, my wife throws things at me a lot. So please, and you can see the way I like the animation if you bring it back. I mean, that's what membranes look like. Membranes are vast. The channels are everywhere. You can see the phospholipid bilayer. The proteins are they're overwhelmingly large comparatively. So look at those things. Thank you very much. We'll go back. We're going to go backwards. Those I don't go for. Primary and secondary. Here you are. Sodium potassium ATPase is another name for it. I just call it, and again, pump and active transport is synonymous. All plasma membranes, but especially active in excitable cells, nerve and muscle cells. That's what makes them excitable. They can change this dramatically, and that way they can conduct signals from point A to point B. That's what this is all about. And we'll get into the directionality, and we look at muscles, and we look at nerves. It has muscles kind of go in all directions. Nerves only go in one direction. We talk about that signal, and then you begin to understand the way things are transmitted. This is the first real glimpse, I think, of really people begin to develop as complex physiology. Aren't you happy you're here? I'm so happy everybody's here. On the top 10 of worst Steeler wins, I mean, that's in the top 10 of ugly Steeler, and they're no team has more ugly wins than they do. It was a pretty ugly win for Tampa yesterday. Moving on. So they're just showing how it works, and this is the other little neat part about that. Maybe you didn't see. Okay, I'll go back and see if I can. It's a 3-to-2 kind of a thing. Remember, there's always more sodium than potassium, but you're going to see this relationship all the time. When sodiums are being effectively, when they're pushing the sodiums back out, it goes with 3 of them. There's 3 sodiums getting pushed back out. There's 2 potassiums being pulled back in. That 3-to-2 relationship is very important. Effectively, think of it this way. You go back to the periodic table. Sodium is smaller because it's higher up on the table. It's a lower number. If you look at the table that's there. So potassium is a larger molecule. And these pumps, it's about size as well as shape that are there. So every time you can move 3, and this is one of those shared ones that are there, at least the way they diagram it, I think it's an easier way to remember it. You have 3 spaces for sodium, you have 2 for potassium. When we get to how muscles work, when we show how muscles fire, you're going to see it goes in the other direction. Okay? We're going to be moving, effectively, we're going to be moving sodiums in and creating that very significant change in the membrane potential and causing eventually a muscle to fire. So you're going to build on this the whole time. And so you can look at this and blah, 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 how it does and phosphorylation, but look at that 3-to-2 relationship and remember it. Secondary active transport is very dependent on that gradient that was created by the primary transport. So they indirectly, low sodium concentration, sodium potassium pump, so there now sodium wants to come in and it can drag other molecules to it, particularly these sugars and amino acids. You'll see that very, very well, and you'll see that a lot in digestive when you get there, and you'll see it going in different directions when you look at urinary. So this is a major building block for AP2. Here's the 3-to-2 thing, blah, 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 blah. So you can see those little places. And so it's diagrammatically interesting, and they're showing it. Now, as sodium is being brought in, here you can see the typical hexagonal hexo shape. It's bringing glucose along for the party. Voila. The last part of active transport, vesicular transport. The vast majority of the way we bring things into cells and spit things out of cells is by way of what are called vesicles. A vesicle is one of those terms you have to use context. Okay, the fancy name for a blister, vesicle. Bless you. If you didn't know that. So we use a lot of terms in anatomy, physiology, and medicine for different things. So doctors will, believe it or not, you have vesicles are like a regular sized blister. If it gets a little bit bigger, we call it, I'm going to try a bulla. And so there's different names. I'm trying to recall back to my ancient, when we used to describe these things, whether it was a vesicle or a bulla. We do all of those kinds of things. It's like if somebody gets a little rash, we describe it one way. And then if it gets a little larger, we go another way. And so that there's a lot of terminology, always no context. Like you probably had that in anatomy. When you're talking about cervix, it means neck. So typically, if you're studying vertebrae, cervical vertebrae. If you're studying obstetrics and gynecology, it's the uterine cervix, which is the area between the vagina and the uterus, which is a neck-like structure. Okay, so what? You're reading questions on tests. Be very observant of the way they are worded. Okay, so voila, it usually is probably the wrong term. It almost inevitably requires energy. So whether we're bringing stuff in or pulling stuff out, it's the way your cells bring in, effectively they eat and drink, and they expel or they waste products. So when you're bringing it in, and remember, this gets back to all those membrane-bound organelles. When the cell brings something in, the potential for harm is there as well as for good. So what do we do when we bring something in? We protect it in membrane. When we've assembled something in the cell, like insulin, like growth hormone, we spit it out, but we protect it during its journey through the cell by membrane. So it's always this continuous idea of membrane, and as you will come to learn in a subsequent lecture, the endomembrane system. So when you endocytose, you're bringing it into the cell. Okay, endo for bringing it in. Cytosis means the cell. If it's food, phagocytosis, solids. If it's oils, pinocytosis, or liquids. And we see both of those. Sometimes it requires signals made by receptors to create, as you will see, this sort of invagination or indentation, as the cell has this miraculous ability to change shape. So, when we reverse it with something called exocytosis, exiting the cell. You even see some, again, when you get into urinary and digestive, sometimes we just transit things through the cell. Generally, you'll see when we bring something into a cell, we process it. Because you don't know if it's a good guy or a bad guy. It might be laden with bacteria or viruses. So every time you're breathing in, everybody, you're breathing in viruses. I don't know if my lectures are boring, but nobody's collapsing just yet. Thank God. One person, God bless him, smiles in the whole class. 2. I'm in a good mood. Only because the Steelers won and the Chiefs didn't lose, so I'm alive in my fantasy pools. in my survivor pools. And I was going to put it all on Buffalo. Buffalo almost lost. Are you kidding me? You can't figure out this effing league. But I digress again. Nevertheless, transcytosis means it's just transiting through the cell. Vesicular trafficking, transporting from one area of the organelle in a cell to another. We'll study this in the endomembrane. So endocytosis. The vesicles, all of them have embedded, they're a membrane. So they're going to have embedded proteins. Very selective in nature. Okay, it's got to be able to bind and be pulled in by the receptor in that part of the membrane. And this is part of the problem. Pathogens, disease-causing things like viruses or bacteria have the ability to bring things in. I mean, most famously HIV. HIV basically it's almost like a look-alike phenomena where one of your macrophages brings this in and really starts this terrible process. And as what I was saying before, when we are always inhaling these things, our body's defenses cover most of this. We inhale viruses and bacteria all the time. You can't help it. Once the vesicle is pulled into the cell, it's either going to go right through the cell into another structure, typically the bloodstream. That's called transcytosis. It transits through, but all the time. And this is really how your defensive system works. Okay? It basically fuses with something called the lysosome. When we get to that part of the cell, the lysosome is another vesicle filled with chemicals for digestion and defense. If you're gobbling up something good, that's nutritious, those enzymes in there will digest it into the appropriate components so you can basically benefit by it, do energy transformations, building blocks, whatever. If there are bad guys in there, it will destroy them. And then you'll spit out, effectively by exocytosis, the waste material. So you're going to see this has some very important language. So here's one of these coded areas. Okay, here's a substance coming in. Those proteins eventually are going to detach. Okay, and you don't have to go crazy about that. But when you finally get to here that now that is uncoated, you have brought in some material by an endocytic vesicle. It could be solid, phagocytosis, or liquid. It's going to bond, okay, with a lysosome. Here's the lysosome. When they bond together, that lysosome will release particles for digestion or defense. It'll either expel waste materials or it'll be utilized inside the cell. It's not the best illustration. We have a fancy term for this. It's called a phagolysosome. That's the way your body deals with it. You have white blood cells called macrophages, big eaters, the largest of the white blood cells that are constantly at it. And they're in your bloodstream, they're in your tissues, and what they do is they latch on to materials that have been tagged by our immune system. This stuff is bad for you. We've isolated it, we've neutralized it, we gobble it up, and we expel it. The macrophage, basically, this is how a white blood cell works. This is how every cell works. Anything that comes into the cell is subject to this. Okay, if it's going to be broken down in any way, shape, or form in the cell. Other than transcytosis, where it's just going completely through. This is really not an example of it that's there. It's always going to be subject to fusing with a lysosome. It's got a fancy term called phagolysosome. And then effectively we get rid of the waste material. You remember me talking to you about Tay-Sachs. The problem is the waste material we couldn't get rid of it effectively. And they'll show you through all of these. I'm not worried about the proteins detaching and blah, 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 blah. Another term we call it is an endosome, okay? So sometimes you can just be going out, be transiting through potentially. So endocytosis is referred to cell eating because it implies solids. And what happens is, as you're going to learn inside the cell, we have very, very neat things called microfilaments. What they can do fairly easily. Oh, my don't have a poop bag. I don't know what's happening in life. But effectively, we'll use paper towel. It's almost like you can take, like, use the shirt if I wanted to. Yeah. Effectively, I'll bring it in next time. Here's when it gobbles something up. So it comes to it, and a little finger inside the cell and push protrusions, gobbles it up, brings it back in. I like it. Very good. Who doesn't like a cell? And even better yet, in your immune system, it'll take something like this in, and it's called an antigen presenting cell. For bad guys, it swallows it, and it presents a part that your immune system's going to form antibodies to. On top. That's the way your body works. It's miraculous. Okay? So it forms this thing, membrane protections, false feet, pseudo for false, pod for feet, form and engulf it, around it. It's also how, if you study this and you looked in pond water, you learned about amoebas. We call it amoeboid motion. It's my 4-month-old great-grandson we were watching is getting very close to that. He's near into scooch. We're going to have to baby-proof. You can see he's, sooner or later, he's going to be down on the ground and scooching around, crawling, can't be far behind. And so, effectively, that's amoeboid motion. Okay. So it's just sort of scooching around, okay, and effectively, it's almost like you see material streaming and pushing the cell this way because it's flexible. Okay, so phagocytosis is something by macrophages, other white blood cells use it. Cytoplasm flows into those temporary extensions, it moves around, and eventually it begins to push on these areas. The receptors are able to trigger and then subsequently engulf this. Pinocytosis is cell drinking because it's in the fluid phase, etc. We do a lot of environmental sampling because we're looking for bad guys. That's what your immune system, I was talking about those antigen presenting cells, they're everywhere. You learned about it with skin, okay? You have keratinocytes, melanocytes, okay? Merkel cells, and you had those Langerhans cells. Basically, they're sampling the outer environment. See? Putting it all together. And you're going to see membrane is conserved. I don't want to give that short shift. So I'm trying to hustle and get this in, so we'll call it a lecture.